PGC reduces CSF melatonin levels and dampens its rhythm, resulting in chronological disturbances, including insomnia and migraine. The low levels of CSF melatonin also elevate neuronal damage from reactive oxygen species, thus accelerating the progression of neurodegenerative disorders. It's been reported that the serum and salivary melatonin and its urine metabolite are negatively related to the size of the pineal calcification and positively related to the uncalcified portion of the gland. In Alzheimer's disease, patients had a higher portion of calcified pineal glands and a lower portion of uncalcified glands than patients with other dementias. This suggests that CSF melatonin levels in Alzheimer's patients would also be significantly reduced, a theory supported by postmortem CSF analysis from these patients, which showed low levels of melatonin. This reduction in CSF melatonin levels is an early event in the development of Alzheimer's disease prior to the manifestation of clinical symptoms. PGC is also associated with aging, even though it has been detected in neonates or children. The incidence of visible PGC increases with age, with rates as high as 83% in individuals over 30 years of age. In some animal species, the incidence of PGC reaches 100% in advanced age. This suggests that PGC is an inevitable process of aging in vertebrates, and slowing down this process may retard the aging process. Despite PGC being a widespread phenomenon in vertebrates, its importance has been ignored and little attention has been given to this issue for decades. Currently, little is known about its exact formation processes and mechanisms. It seems that there are two origins of PGC, in association with pinealocytes or with non-pinealocytes. Some studies found that PGC was restricted to the connective tissue, representing aging-related calcium accumulation within this tissue. This type of calcification is similar to that found in other brain structures. High initiation of PGC has been associated with tryptase-containing mast cells, which release biologically active substances, including tryptase, that participate in calcification. This process is pathological, but not age-related since it also occurs in children. The formation of PGC seems to have two origins, one associated with pinealocytes and the other with non-pinealocytes. Some studies have found that PGC is restricted to the connective tissue, involving the formation of calcareous deposits within the connective tissue stroma of the gland. These deposits represent aging-related calcium accumulation within the connective tissue, a type of calcification similar to that found in the habenular commissure and choroid plexus. The connective tissue-derived PGC is predominant in rats and purebred white guinea pigs. In the analysis of human pineal gland specimens, it was reported that the initiation of PGC was associated with tryptase-containing mast cells. During systemic or local pathological conditions, these mast cells infiltrate into the pineal gland, releasing biologically active substances, including tryptase, which participates in calcification. This process is pathological and not age-related, as it also occurs in children. The incidence of visible PGC increases with age, with rates of 2% at ages 0 to 9, 32% at ages 10 to 19, 53% at ages 20 to 29, and 83% in individuals over 30. This clearly indicates that the degree of PGC increases with aging. In some animals like turkeys and rats, the incidence of PGC reaches 100% in advanced age, suggesting that PGC is an inevitable process of aging in vertebrates. Therefore, slowing down this process might retard the aging process. In Alzheimer's disease, patients had a higher portion of calcified pineal glands and a lower portion of uncalcified glands compared to patients with other dementias. The serum melatonin rhythm resembles the CSF melatonin rhythm, indicating that CSF melatonin levels in Alzheimer's patients would also be significantly reduced. This reduction might be an early event in the development of Alzheimer's disease, possibly occurring even before the clinical symptoms manifest. If these patients had PGC, their CSF melatonin levels might further decrease, accelerating the disease process. It has been reported that serum and salivary melatonin levels and its urine metabolite are negatively related to the size of the pineal calcification and positively related to the uncalcified portion of the gland. The post-mortem CSF from Alzheimer's patients showed low levels of melatonin, only 20% of that in non-Alzheimer's controls. This suggests a potential role of PGC in the development and progression of Alzheimer's disease. There are two primary theories regarding the origin of pineal gland calcification from pinealocytes. One theory, proposed by Lukasik and Reiter, suggests that pinealocytes release polypeptides into the extracellular space, which actively exchange with calcium to form a calcium carrier complex. This complex is then deposited on the surface of adjacent multilayered concretions, linking the formation of concretions to the secretory function of the pineal gland. Another theory, proposed by Kristic, suggests that the initial sites of intracellular calcification are the cytoplasmic matrix, vacuoles, mitochondria, and the endoplasmic reticulum of large clear pinealocytes. These sites transform into a cervuli by further addition of hydroxyapatite crystals, leading to cell degeneration and death, and eventually releasing the acervuli into the extracellular space. High intracellular calcium levels could be a trigger for this process, possibly due to the failure of calcium APase, a phenomenon linked to aging or pathological conditions. The authors propose a new hypothesis suggesting that PGC might be an active process rather than a passive one. They speculate that the pineal gland has a blood filtration function similar to the kidney, given the similarities in vascular structures and blood flow rates between the two organs. However, the composition of PGC differs significantly from kidney stones, with PGC primarily consisting of hydroxyapatite, a chief structural element of vertebrate bone. The formation of hydroxyapatite is believed to be a programmed process involving collaboration between bone cells, resembling a physiological rather than pathological ossification type with characteristics between enamel and dentine. Recent studies have shown that melatonin enhances the ability of mesenchymal stem cells to differentiate into osteoblast-like cells both in vivo and in vitro. SCs found in the early stages of pineal development in birds and rats play a crucial role in pineal follicular formation later during gland development. These cells can differentiate into various cell types, including muscle cells and potentially osteoblasts and osteocytes. This differentiation seems to be melatonin-dependent, mediated through the melatonin membrane receptor 2. Melatonin binds to the MT2 receptors on MSCs, promoting their differentiation into pre-osteoblasts and further facilitating osteoblast proliferation, osteocyte formation, mineralization, and bone formation. The pineal gland has a complex vascular system, making it vulnerable to chronic vascular inflammation during aging or certain disorders. This inflammation can mobilize the migration and adhesion of mesenchymal stem cells in the gland or promote their proliferation due to increased levels of pro-inflammatory cytokines. The interaction between vascular MSCs and inflammatory mediators, especially interleukin-22, leads to MSC's proliferation, migration, and osteogenic differentiation under the influence of high levels of pineal melatonin, eventually resulting in PGC formation. Various pathological conditions can cause brain tissue hypoxia, including hypertension, sleep apnea, stroke, and respiratory disorders. Hypoxia-inducible factor 1 alpha promotes the proliferation, migration, and adhesion of MSCs in hypoxic areas, including the pineal gland. Generally, hypoxia increases bone resorption and suppresses osteoblastic differentiation and bone formation. However, in the pineal gland, melatonin promotes osteoblast differentiation and mineralization of MSCs under hypoxic conditions, favoring PGC formation. Some cells of the pineal gland are influenced by intracranial pressure as they swim in the third ventricle. Increased intracranial pressure, associated with various cerebral disorders, can impede the pineal filtration rate and induce endoepithelial cell damage by chronoinflammation, promoting bone remodeling and mineralization, thus facilitating PGC formation. Researchers have attempted to rejuvenate the pineal gland through ectopic transplantation. Initial studies found that transplanted pineal glands in rats were innervated by surrounding sympathetic nerve endings, establishing a normal rhythm in AA and NAT activity similar to the in situ pineal gland. However, except in the anterior chamber of the eye, no melatonin circadian rhythm was detected in transplanted glands, and melatonin levels could not match those produced by the in situ pineal gland. The lack of melatonin rhythm may be due to the absence of sympathetic innervation in the grafted gland at sites other than the anterior chamber of the eye. Functional studies of ectopic pineal gland grafts have shown promising results. Transplantation of young pineal glands into the thymus of older mice partially prevented thymic involution due to anti-apoptotic activity, preserved age-related alterations in erythrocyte membranes, and even extended the lifespan of recipients by up to 27% compared to controls. This life extension effect is attributed to the high levels of nocturnal melatonin released by the grafted pineal gland, rejuvenating the thymus and preserving the immune responsiveness of older animals to levels seen in younger animals. Ectopic transplantation of the pineal gland seems to have several health benefits. However, it cannot fully replicate the functions of the in situ pineal gland, which secretes high levels of melatonin directly into the cerebrospinal fluid, protecting the brain from oxidative damage. This gland also exhibits a circadian rhythm, especially in the CSF of the third ventricle, where the nighttime melatonin peak has a sharp rise and fall compared to its serum circadian pattern. This alteration in CSF melatonin is believed to serve as a signal for the biorhythm of organisms. The ectopic pineal gland transplants lack these two critical aspects. Therefore, a more effective method might be transplanting the pineal gland into the pineal region or the third ventricle. Some studies have shown that pineal glands transplanted into these areas survived due to revascularization and partial reinnervation, producing melatonin, albeit at lower levels and without the nighttime rise. To maintain a healthy and functional pineal gland, efforts should be directed towards slowing its calcification or restoring its functions. Several pathological conditions can accelerate pineal calcification, but environmental biohazards, such as fluoride pollution, also contribute. For instance, the pineal gland in goosanders concentrates fluoride, a water pollutant, at levels 5 times higher than in the brain. Similarly, high fluoride levels in the human pineal gland are positively associated with calcium accumulation in the gland. Reducing environmental fluoride pollution might help delay or prevent premature pineal calcification. Increasing the availability of calcium salt crystallization inhibitors like pyrophosphate and phytate protects against pathological pineal calcification. Decalcifying the pineal gland is possible using methods like pineal microbialysis, which is commonly used to measure the gland's melatonin production. This method can be employed to decalcify the gland using EDTA or acidic solutions as eluents capable of dissolving calcium deposits and removing them through dialysis. Additionally, cells isolated from young pineal glands or engineered stem pinealocytes could be directly injected into the decalcified in situ pineal gland. These cells have a high chance of survival in the gland due to the melatonin levels generated by the gland, promoting the survival and differentiation of transplanted stem cells in various organs and tissues. Studies show that injecting cells collected from the pineal gland of 1-day-old chicks into the in situ pineal gland of 4.5 to 5-year-old hens improved egg-laying rates and the general well-being of the older recipients, indicating a first step towards rejuvenating calcified pineal glands to maintain optimal human health. The article concludes by emphasizing the potential methods to rejuvenate or maintain the optimal functionality of the pineal gland, which is known to undergo calcification, a process associated with several health issues including neurodegenerative diseases and sleep disorders. The authors highlight the promising avenues in the field of pineal gland research, particularly focusing on the potential of decalcification and rejuvenation of the gland. He authors propose that decalcification of the pineal gland may not be an impossible feat. Current techniques like pineal microdialysis, which is frequently used to measure melatonin production in the gland, could potentially be employed to decalcify the gland using substances like EDTA or acidic solutions as eluents. These substances have the ability to dissolve calcium deposits and